Okay, good morning. We'll go ahead and get started. We have a pretty full day today. So we have three separate components of this session today. We'll start with a discussion of good clinical practice, which is really a series of guidelines for how to make sure that clinical research is done in the way that makes it most efficient, most safe, most ethical, the whole picture. So it's in this section of ethics because it's really a major part of it is to protect the clinical subjects, but it also has a really strong focus on making sure that the research effort is as effective as possible, and that the funds that are used to do the research are spent effectively and wisely. We'll also be talking about biorepositories and some of the opportunities there are on our campus to have samples processed through a sort of contract mechanism for you. And then in the last half hour, it'll be somewhere between 15 and 30 minutes. We'll talk about your challenge projects and the general way of thinking about these, and we'll go through what you wrote and talk about sort of the process and ways to maybe do this more effectively and to refine your ideas as we go forward. So the first part is going to be led by Meredith Fitzgerald, who is the director of a component of the university's research support effort that's called the Clinical Research Support Group, or CRISP. It's linked to the CCTS, but it's really open to everybody around campus, and Meredith will probably give you a little bit of information about some of the services they do besides the GCP as she goes along, to give you a sense of what kind of things they can really do for you. Meredith is the director of that unit, and she's going to work this off. Thank you.
I'm going to tell you a little bit about my background and then what we do, and then if you have questions or want to add, I really highly encourage. GCPs can be not the most interesting, but it's the most important to me topic when you're doing research. It's basically the building blocks. So if you have good clinical practices and you incorporate that in everything you do, then your research will be successful. So I am a nurse, and I was a coordinator for 9 years before I joined the Clinical Research Support Unit. And there was a need across campus that Penny Jester, who I've adopted these slides from and who just recently retired, started this unit. And what we started off with was 2 employees. Basically, we went around, and if you needed a coordinator, we provided those services. And then it morphed into data management, specimen management, any monitoring, anything that you possibly could think of, we provide in education. And a big part of our program is providing educational programs to everybody across campus. And we will go through, we do the research training program, investigator training program, and then research seminars are on the first and third Thursdays of every month. We're always looking for topics, so if you have any suggestions, please email us, call us. We're available to you. Even if you don't need our services and you have questions and you want to link to somebody across campus, we can help you find what you need. So that's just a little bit about us. So what are GCPs and why are they important? This is things that we're going to cover today. And when are they implemented and who implements them? So what do good clinical practices address? Study implementation. So from the time that you may think of a project or have a thought, hypothesis, to the time that you're closing the study, GCPs should be incorporated in every part of that. And it provides guidelines to assure the rights of subject, that study subjects are protected, and it assures that the information that you collect is accurate and reliable. So research, you probably already know this, is systematic. Investigation including research development, testing, evaluation, and it's to develop or contribute to generalizable information and knowledge. So the International Conference on Harmonization is a regulation body that's helped incorporate to define standards on how to practice research. And one of your handouts has 13 principles. And I really encourage you to keep these 13 principles and really read through them and apply them in everything that you do. They're very basic, common sense kind of things, but you would be surprised what we see across campus and how investigators don't intentionally mean to do something, but they just don't know. So these educational programs and these regulations are really important because it helps you guide where you do the research correct and that it's valid. So if you have time, later on in the slide set, the ICH has regulations, and it breaks it down, and GCPs are applied to all these regulations. We're not going to go through all of it, but it's a good reference to see where it's incorporated. So the objectives of the ICH GCPs is to provide unified standard applications that can go across all different types of research. It facilitates mutual acceptance of clinical data, and it was developed in accordance with multiple countries and the World Health Organization. So there's different sources of GCPs, and these are all the sources. You probably have already covered this in your previous classes, but I will say that I keep and I review regularly, especially the ICH GCPs, the 13 principles here, the Code of Federal Regulations. If you ever want some really boring material to go to sleep, read it. But I will tell you, over the 16 years of doing research, if I'm ever in a situation and I need to refer back to them, I keep an app on my phone, and it's always good to go back and review. Goals of GCPs and goal of research basically is to protect human subjects and to assure that you have accurate and reliable data collection. The impact is that it strengthens the ethical and scientific standards that, when adhered to, enhance the quality of research. And I hate to say this, but if you don't follow these regulations and you don't follow your protocol, then why are you doing the research? It's so important, and we have so many things that are, black box warnings. Okay, I got it. Those have been issued. We're starting to see more of those. Recalls of drugs. If it was reported correctly the first time, maybe we wouldn't have these things. So it's really important to make sure your records, just the basic AEs, adverse events, SAEs, make sure that we really are doing our jobs and that we're diligent in reporting and documenting it, making sure that our T's are crossed and our I's are dotted. So GCPs are standards for designing, conducting, performing, monitoring, auditing, reporting, analyzing, and reporting data. So as you can tell, it's in every part. It doesn't matter what type of research you're doing. You could be doing behavioral studies. You could be collecting questionnaires on students. It's just important to make sure that in every part, when you're designing protocols, we often see investigator-initiated protocols that aren't thought out all the way through. And there are resources here with the CCTS that can help you design your protocols. But thinking about, from the time you start with the idea to the time you finish, what information you are going to record, why you're going to record it, what you're going to use it for, how you're going to collect the information. It really goes back to doing a really good feasibility. And we'll talk about this when we get into recruiting and retention, but making sure that you really think from the beginning to the end what you're going to do. So where did GCPs come from? Laws, regulations, guidance documents, guidelines, SOPs, and training and compliance manuals. They build this pyramid. It starts from training, audit, compliance manuals, all the way up to the objectives. So that every part of it, SOPs, standard operating procedures, How many of you have SOPs in your office? Do you even know what an SOP is? So SOPs are really important. They tell you exactly what you're going to do. We have SOP templates on the CCTS website, and you can take those templates. We've got a group across campus together, and you can take those templates and modify it to your office. So it doesn't really matter if you're in the School of Nursing, Public Health, or whatever. You can still use those templates and make it your own. But it just kind of keeps everybody on the research team doing the same way of doing things. So why so many regulations? And I understand that you've already talked about the Tuskegee syphilis study and Jesse Gessinger and numerous other tragedies that have happened, but it keeps it systematic. It protects the human subjects, and we just have to stay very cognizant that these things happen, and they still happen today. And that's the thing that we think, oh, this is in the past, but no. I've had investigators look at me and say, can't we just do this and ask for forgiveness later? And that was recently. So you really need to know what your rules are. You need to have things reviewed by the Institutional Review Board. They're there to protect patients. And just think if it was you or your family. You know, I had a nurse just recently that was diagnosed with cancer. And she is enrolled in clinical trials, and she looked at us and she said, I hope the data is valid. I hope somebody did their job. And when you think about it, I still get chill bumps because she's depending on data that we collected, but the research community. So if we don't do our jobs and we're not vigilant and use these 13 principles in our practice, it really could affect our loved ones and even us. So who do they apply to? Everybody. I'm talking from the person that collects your labs to the person that enters your data. The investigator is responsible, and PI does not mean practically invisible. You need to be involved. You need to meet regularly with your research teams. Communication is key here. And if everybody's on the same page and the investigator knows what's going on, and gives feedback to the study team, you're all moving in the same direction. And that's the overall objective of GCPs. If the PI is not available and the team is floundering around and one member thinks that it's okay to do this, another member thinks it's okay, it's not systematic. And if you can imagine, if you don't have documents to record this on, I've walked into offices where literally it was the size of a closet and there were stacks of papers all over the floor. The regulatory binders weren't kept. The patient files were not organized. Patients were enrolled that weren't supposed to be enrolled. We had a major cleanup job. It was a big mess, and it puts the data and the research in question. And that's not what we want to do. And you know, it was probably unintentional, but it happens. So everybody. So the NIH in 2016 decided that all investigators that are doing the NIH studies have to be GCP trained. So if you noticed, or if you were at UAB back then, everybody had to have GCP training, and every 3 years you have to repeat it. So you have a refresher course, which they just developed, which is nice that you don't have to take the initial course again. But it's really important. And so UAB adopted this and said that, you know, all investigators, all research team members that are doing research need to have GCP training. So this is where I was talking the ICH outline of contents and where GCPs are embedded in every part of it. So I'm not going to read this, but I just included it as a resource. So if you wanted to go to a section and read that section, it kind of tells you it's broken into, I believe, 8 parts.
So the ICH regulations are available online?
Yes.
So
yes, it's the International Conference of Harmonization. So the ICH is much easier to remember and say. And also the federal regulations are too. There's an app that you can, I just keep it on my phone. But anyway, we'll just kind of skip through these slides. The differences in required regulations are based on the types of research. I will say device trials are a little bit different. than like a drug study. They're all similar and they all use GCPs in all of them. But like a device trial, you don't sign a 1572, you have a letter of agreement. So there's a little bit, their guidelines and regulations by the FDA are a little more broad. Where drug studies are more streamlined. But if you're familiar or if you have questions, the IRB is a really good resource, and then we can stream you into who you need to talk about for the different guidelines. Even in behavioral research, and we get this question a lot, so this is why we included this, GCPs apply. This is the website, and I believe you had a talk on the IRB on how to do the training, but sometimes it's hard to find it. So if you go under research and you go under research offices and go under institutional review board, there's a site that says training. And under training, you have GCPs. You have the initial course and the refresher course. If you ever have questions or can't find it, please call us. I'll give you our phone number and we can walk you through step by step on how to find it. And this is the website. So we're expanding beyond just GCPs into competencies. And this is something that you'll start seeing in the future. Carolyn Thomas-Jones used to be at UAB and now she's at Ohio State, and she came and talked at our research seminar last week. But she's been instrumental in getting competencies across the United States for researchers. And we're really excited about it because what we would like to do here at UAB, and we're planning on doing this really soon, is develop job titles, research job titles. And basically, if you're a researcher, you would come in, you would fall under this title, and there would be certain competencies that would go with it. So our research team, as you get more experience, you would have more competencies and you would be better trained. So that's coming. So it's basic from GCPs, it will go into competencies and be implemented across the United States. These are the domains, and if you notice, clinical trials, operations, GCPs will be incorporated in those competencies. So I'll let you read through those on your own. These are the offerings we have at UAB. Within 2 weeks of employment, we hope that you've completed the CITI GCP training and HSP training. This is very important. Again, it's on that training website where you go to GCP, the HSP training is there as well. Within 2 months of employment, we have the research orientation program. That's for all researchers across campus to come to. We do it every other month. It's a 4-hour course, and it's just a broad overview. It gives you resources. It's a really good primer. We go through the 13 principles. I do the GCP talk, but it's a really good resource. Then within 6 months of employment, we offer the research training program twice a year and the clinical investigator training program twice a year. And the research training program is a 7-week program, and we break down all components of research. We have speakers from across campus, OSP, Office of Sponsored Programs, IRB. We talk about monitoring, we talk about protocols, how to design a protocol, what are some basic things when you're reading a protocol you really need to pay attention to. It's a really comprehensive class. It goes into a lot more detail than the orientation program. And the clinical investigator training program is just for PIs and investigators, and it's another comprehensive. Dr. Sally, Cindy Joyner, they're all involved in that, and it's a very good program.
As a PI, do you recommend that PIs attend the RTP to see what the staff is learning?
It would be we do have PIs that attend it, and yes. And you could also, we could send you the agenda, and if there's classes that you really are more interested in, then you could come to those classes.
Okay, great. And one more thing, can you provide the name of the app that you said you have downloaded?
Yes,
on your
I will
phone?
find it for
Okay,
you, and I'll
thank
give it to you. I'll give it to Dr. Chapman. And then twice a month, we have continuous training research seminars. And at the seminars, we pick topics that seem to be like the hot topics or updates. I know the common rule is going to be updated in July, and the date hasn't been formally set, but there's been conference calls about that. In the common rule, they will change In the informed consent, there'll be a summary that's required. The IRB will give us guidance to what consent forms will need that summary. They're also trying to write consent forms where patients can understand it. It's not as long. It's more directly to the point, because if you have a 23-page consent form, the patient's by the 14th page is looking at you cross-eyed, and really it's important to go through and really explain. And when you consent a patient, you're not just consenting them one time, you're consenting them through the entire duration of the study. So it's very important that they understand and they continually agree to participate in the study. So that's coming up, and we'll have some educational as the guidelines come out. And the upcoming rules updated, we will have trainings on that. But anything that you need, topics, we can bring speakers in. Just like I said, we did one on quality assurance and QC, having a process and why it's important. And Carolyn did a wonderful job. The slides we'll probably post on the website, but things like that, we're hoping in the future that we'll start recording them, because we know Thursdays at noon, that could be a hard time. You may be in clinic, but we're hoping that we will have more resources that you can watch it on your own time.
Correct me if I'm wrong, but I think the CCTS Digest every week lists the topic of the upcoming research seminar, so you can be reminded about that. Can I just ask, how many people are members of the CCTS group? So I really encourage you to sign up. I mean, the only downside is that you'll get an email every week that's the CCTS Digest. The good upside is that you'll get that and you'll have access to any of the activities that the CCTS runs. So if you just email ccts@uab.edu and ask to join and be included on the mailing list, that'll happen instantly. So I encourage you to do that. So this looks like a lot to me, and to give people a sense of the burden of it. So the city course and the human subjects protection training, my recollection is that each of those, if you're focused on what you're doing, takes anywhere between an hour and an hour and a half.
They're broken into modules. So
they're little modules. Yeah, you can do it a bit at a time. And the continuing training that's required, I always learn stuff doing it and get reminded of things I've sort of forgotten to keep in the front of my mind. The RTP and the clinical investigator training program, is there any cost for those, or are they free?
The research orientation program is free, and RTP, I believe, is $125.
Okay.
There is a cost.
The RTP and the clinical investigator training program, that's a significant time commitment. That's several sessions.
But we understand that you have clinic and you have patients and you have other obligations. So in the 7-week period, if you miss a class or 2 classes, we offer it twice a year. So you could actually come back the following year, just let us know and say, hey, I need to pick up day 3 and day 4. We try to keep the syllabus pretty consistent, so you can always come back. And a lot of times when you send employees, it's overwhelming. It's a lot of information. So if you're new to research, I usually tell employees it's going to take a good solid year to kind of get your feet planted and really understand all the parts. So you may want to come back in a year and repeat certain classes, because it's almost like information overload. So if you break it down, and we try to break it down, it makes more sense and it's more applicable. That makes sense.
Thank you. Is there any funding available to send staff for PIs that have very, very, very lean budgets?
That's a really good question. I'm not going to say no, but I think if you came to us with those situations, our whole mission, the CRIS program, is to improve research across campus. So I don't think we would turn you away. I don't have that authority, but I don't think,
I don't think this has ever been tried before, so I may have my hands slapped later. But so the CCTS has something called research vouchers, which can pay modest amounts to help people access services that the CCTS offers particularly. So it needs to be linked to a research project, and there needs to be justification for why you need to do this. But I've never seen anybody think to ask for the support for the training through that mechanism, but I think that would not be unreasonable to do, and
And there's
we can show you how to access that if you're
Yeah.
interested.
There's a flyer out, or there was a flyer out in the lobby on the voucher program, so you can get
Oh, I've
information.
benefited from the voucher
Okay.
program. Love it.
So I don't think we would turn you away because the whole mission, I mean, like I said, is to improve it. And I can be very honest and bluntly, the trainings are not mandatory, but the groups that participate in the training, their research is much better than groups that need the training and don't come. So it's worth, yes, it is a time investment, but in the end, it's worth. You're up with what's changing, what's coming, and research is constantly evolving. When I first started 15 years ago to what it is today, we didn't have these training programs. And I will tell you, I learned by mistake, sadly. I had an excellent PI, and she would laugh, and she'd say, I don't look good in black and white stripes. Well, then, the prisons changed to orange, and I would retort back, well, I don't look good in orange. But we kept each other straight. And when in question, we always called and asked. The IRB probably saw my phone number and go, great, she's calling again. But it really, we have resources across campus, and I would much rather you ask questions and not be that PI, can't we ask for forgiveness later? I mean, that's not the best way to approach things. We write protocols and we follow protocols for a reason. We want it to be systematic. We want to be able to reproduce it. And I will say that in the different areas I've gone to, I've seen protocols that we haven't thought all the way through, and those are the ones that cost multiple amendments, which we did a paper that was published. It cost a lot of money to do those amendments. So if you really sit down at the beginning and really plan, get some input from your team, can this be done? Does this make sense? As simple as doing a urine pregnancy test for adolescents in a study. I had an investigator says, do you really think that patient's pregnant? And I retorted back, have you seen Dateline? How do I know that that patient is not pregnant until I do the test? And adolescents don't urinate on demand. So maybe it's worth doing a deviation and worth writing, that's a minor deviation, but it's a good deviation because you're looking at patient safety. And we can do a serum pregnancy test because they've already had labs drawn. And all it is a simple added lab test to it, and then you prove that that patient isn't pregnant. And the FDA had that pregnancy test put in that protocol for a reason. So it's just simple things like that, thinking it all the way through. It really is not a hard step, or it didn't add a whole lot.
Question. So does the RTP allow for someone who doesn't have a medical background, a nurse or a doctor, to be credentialed? And I ask this because I've been a study coordinator who's not a nurse, and she's not allowed to enter any impact orders. So they said you need to be credentialed.
We have tons of non-nurse coordinators out there across campus. And you're right, there's a policy out there that non-nurses can't enter orders into impact. But yes, they can come to all the classes, and they conduct a lot of our research that's done on campus.
So is that a set rule that non-nurses cannot enter orders on impact? Because I know of some study coordinators who can, and they're not nurses. I mean, I have two coordinators, and one can, the other cannot. So
this just recently came up, and there's been a lot of meetings. Brian Bates just talked, and I forgot to mention the lunch and learns that we have quarterly. They're campus updates, so those are really good. We advertise those and send emails out. But Brian Bates at the last lunch and learn in January talked about this, and apparently it's not widely known, but it will be implemented more. And as we move into Encore, which is a medical database that it's helping us maintain our research at UAB, there's a thing called Power Plans. And Power Plans is what flags your patients and impact if they're in a clinical trial, and it's what you enter orders for labs and radiology. It will expand to more, but in order to get access to do that, you have to be a licensed personnel. And that goes back to that policy. So it is going to impact those non-nurses who used to be able to do it won't be able to do it. And there'll be more. We know it's going to be a huge issue, but there'll be more on that. And the policy will be circulated because apparently the policy has been there for a while. It's just that it's not well known. Does that help?
Yes, thank you.
So one of the things we try to do through this program is to help make sure that people who are joining us from partner institutions for the CCTS recognize things that they can access from their institution that will be relevant to them that aren't going to be specific. And also, I realize one of the things we ought to be doing when we do this kind of a session is really have collated complementary programs that may exist in the other institutions so we can help you access those. And I'm not sure that we're prepared to do that right at the moment, but that's something we'll put our heads around and try to create some links on our website that will help make sure that people can access similar kinds of activities at the partners. But Meredith, maybe you can just comment on which of the aspects of this would be totally appropriate and relevant to people regardless of where they're from, and which things are either only accessible at UAB or are really particularly only relevant to people within our system here.
I would say with the research seminars, we try to keep those and we broadcast the ones that are generalizable. If they're UAB specific, it may not be everything they can use, but then they can take ideas from those things and apply it to their institution because they know how their institutions are set up and what they can do. The research training program is very generalizable. The investigator training program is too, so I think both of those would be really beneficial for everybody to participate in. And we do have plans hopefully in the future to get this where it's more available to the partner network. Okay, so I've got to go on speed dial because I see Jeff's here and I've got about 20-30 minutes left. So clinicaltrials.gov. How is this all tied in with GCPs? Well, if you know, clinicaltrials.gov is for the public. It's a place where the public can go and look at all the clinical trials that are out there. So we owe it to the volunteers for the trials to have access to knowing what the results are. So this is a place that you enter your trial information, your protocol information, and then the public has access to it. And we do get phone calls from people that are looking for trials. They can, your investigator name the institution, they will call and say, I want to participate in this. So it is used. So we won't go into all the history, but I've included it in your slides so you can understand why it was involved. The most important thing about this is in 2016, the final rule came out and that they're mandating clinical trials, certain clinical trials. And if you have a question, if your clinical trial belongs in clinicaltrials.gov, we have a wonderful resource on campus that can help you. They can help you get the information in the website and maintain it. But the most important thing is they're starting to implement fines if your clinical trials are not entered into the database. So this just goes into the rules. We'll kind of skip through this. And then the Department of Health and Human Services also came in and said these are the reasons why, and this is what we're looking for on the website. So I won't go into all the details for sake of time, but if you've ever gone to the website, it's very user-friendly. We can help you step by step. They will come in and query, and we get a report of how many queries, and we have somebody that will contact you because you have to answer those queries within so many days. And so Denise McKenzie is the contact person with the CCTS, and I will give you her phone number, and her information is on one of the slides. She's an excellent resource. She will help you walk through it. She will come meet with you. She will, they want outcomes, they want reports each year, and then when you get your final results, you have to put those, and there's a certain way to do it, and she is here to help you do that. So all protocols that are part of the NIH have to be entered into clinicaltrials.gov. And then this kind of goes into and breaks it down to what trials need to go in and don't. It is always being updated, and they're going to require updated requirements to be entered into clinicaltrials.gov. Denise is doing a clinicaltrials.gov workshop on the 22nd. She does this regularly. She picks a topic or an area and she goes through it, and she will walk you through it. If you don't meet with her person to person, you can send your team, because you can, as a PI, delegate who enters the information into clinicaltrials.gov. The reports reporting has to be reported within 12 months of the last subject that meets the primary outcome measure. So when is it completed? These are your timelines. And Denise, like I said, we get a report and she will prompt you to stay within. They are starting to fine us. The university is making sure that we are not fined. To date, knock on wood, we have not been fined. Maybe this is more wood. But it's real important to stay within those guidelines, because they have come in and said in 2016, we will start fining institutions and investigators if they don't report per the guidelines. This is just more about reporting. And then the penalties, we can go through that. Y'all can read that. And then you can see the penalties are quite stout, $10,000. And then each day that you don't enter the information, it goes up. So it's just real important to be compliant. Challenges that the principal investigator, we know you're all busy, is held responsible for registering and reporting it, but you can delegate it to staff members. You just have to make sure your staff members are reporting what you want to report in at the website. And then we've gone through that. These are the services we can provide: one-on-one training, phone calls. Denise is always on the phone with somebody. She will prompt you to get those queries. They kind of word things a little bit different, but she can help you interpret what they're asking for. And this is her email address. The office number is 975-2758, and when I update these slides, I will include that. But if you call the CCTS, they can even direct us. The CCTS website, email address is right here. If you have questions, they'll forward those questions to us, and we will answer it. Or you can call me, and I can direct you to Denise. So there's multiple ways of getting in touch and getting help. These are references for the slides. Now we're going to go into study recruitment and retention, my favorite part, because this is when you actually do it. So why is recruitment important? After you recruit, now what? Okay, you've got the patients in there, but what do I do with them? And why is it important to keep them engaged and involved? And what are ways to recruit? And how do we improve retention? So I adapted this slide from Rachel, who had a wonderful set of slides, recruitment and retention. But I thought these two statements were really, really important, because if you do your feasibility and you really think about the subjects you're going to recruit for that study, and you recruit the right patients, they'll stay engaged in that study. If you pick poor and you don't really screen them out, or if you hear a patient say, I have a sick loved one, I don't know if I can make it to you already hear barriers before you've enrolled them, they're not going to be compliant. And then you've got them in the study, and now what do you do with them? So it's really important from the very beginning to pick the right patients, follow your inclusion and exclusion criteria, and really think through the process. So, recruitment. Know your population. Had an investigator not too long ago that said that they could get 300 patients in this trial. The population was an adolescent, 12 to 20 years old. They had to take 5 pills a day, and they had to be 75% compliance. Now, 12 to 20 years old, taking 5 pills a day, and they have to be 75% compliant. Let's think about that. They had to come in for monthly visits. So that means if they played sports, or they were in school, or they were involved, or in college, that's a lot to ask. So you're going to get 300 patients. Now, he may have 300 patients. I'm not doubting that at all. But let's realistically think, how many patients will really commit to this and follow through? And this is an important trial. It can really impact medicine in this particular area. So we got it down to, I want to say 30. And I still feel like that's a little too high. And I looked at the study team and I was like, do you really think you can recruit 30 patients? I would much rather be realistic, set a goal that I can attain, and then if I can go over that goal, you can always amend your contract and go over it. And then you look like a rock star, you know. So to me, I want to obtain what I set out to do, and I want to maintain those patients throughout the duration of the trial. I want to engage them. I want to form a relationship at the very beginning. I had a 5-year trial, had 60 patients enrolled. We ended up having 5 patients transfer from Louisiana because of Hurricane Katrina. So I had 65 patients, and we kept all 65 in that trial for 5 years. And it was a job. And it was a job in that we had one patient that became homeless, and we had to follow him. But we found ways. We brainstormed. We got together as a team. I got the fellows involved, so if this patient was admitted on the floor, they let us know. I found out places that he worked, and I got permission, and they would call me, hey, he's here. You want to do a phone call? So it's just thinking outside the box to keep them engaged. One of the transfer patients did not sign the informed consent for UAB. I could verify that he was alive, but since he didn't consent to UAB for us following him, I couldn't collect any information. But he was displaced and he moved around a lot, so every time I tried to send in the informed consent, he wasn't there. But getting a contact list with alternate numbers of people that don't live with you was really crucial, because phone numbers are disconnected, but usually a patient will have somebody that will know where they are. And so it may be a sister, it may be a mother, but just collecting that information keeps them engaged, and you can retain them through the whole trial. And then you prove what you're trying to do. You may not be 100% compliant in that you may miss some visits, but re-engage them, keep them involved. Doing that feasibility assessment at the very beginning, thinking, would I let myself or a family member, a loved one, do this trial? And then asking what you're asking the participants to do, and how much time it's going to take them. Continually being grateful for them giving their time. And then how much is it going to cost you and the patient? How can we alleviate those barriers and keep it from costing them money and time? I mean, we can compensate a little bit, but you don't want to, but making sure that you're really cognizant and thankful of what they're giving to you. So how can you contact your population? Now today we have social media, Facebook, radio, television, the UAB reporter, the online service at TKC, so when they're calling to make an appointment, there's a recording that can tell you about different trials. And it's free, or it was. The newspaper ads, websites, a lot of patients will go to these websites. A lot of studies are providing information about the disease process in the websites, and that's really good. Mass mailing, community churches, or community outreach churches, health fairs, different organizations. If you're doing an Alzheimer's, there's Alzheimer's support groups that you could go to. Physician referrals, and then having recruitment materials, brochures, flyers, cards, videos, and waiting rooms. Those are all good ways to recruit. So successful recruiting is reaching your recruitment goal, staying within your budget, following the protocol, and retaining the participants throughout the duration of the study. And then retention, we kind of touched base on this, but really keeping them engaged is very, very important. Knowing your patients. Had a patient one time that always had a toothpick in his mouth. If he had a toothpick in his mouth, it was a good day. If he didn't have the toothpick, it was a bad day. Picking up on those nonverbal clues. Had another patient, he was just jovial. He would walk in a room, big smile on his face, it would light up the whole room. And then one day he walked in, didn't have a smile on his face. Color was kind of gray and ashen. And I was like, what's going on with him? And I looked at him and said, tell me what's going on. He goes, I'm fine, I'm fine. I'm like, really? You don't look fine. As he sat down, I noticed he was short of breath. I noticed that he had this glistening diaphoretic, and I said, you know what, we're going to do a 12-lead EKG. So the study visit went from not doing the study visit to he ended up in the cath lab. But you know, it's those nonverbal clues. We did the study visit right before he was discharged, but that's the added bonus of coming in for a study visit, knowing your patients, being engaged, and really caring about them. So no, that wasn't the research visit, but he could have died. So just keeping them engaged. Giving them calendars, appointment cards, text messages are a big thing, reminding them of their appointments, birthday cards, newsletters, all of those things. Personal contact, I can't stress it enough, and continually thanking them. Be creative, get your research team together, and brainstorm ways to avoid barriers. And if they're lost, I'm sorry.
I had a question. Can you go back to the last slide, please?
I can try.
When it comes to continual personal contact, does that information have to be in the protocol?
I would put it in the protocol. You can always amend your protocol. You can always ask if you start seeing them drop off. That's really important.
Even the thank you letters at the end of the study?
All materials that you use have to be approved by the IRB. So anything that you give to a patient, rule of thumb, and when in doubt, I would call the IRB.
Thank you.
But appointment cards, all of that have to be approved. Any others? Also, like the homeless patient I was talking about, re-engaging him. No, we were not compliant. We did not hit every visit, but we hit enough visits. We were able to collect information. We were able to send that information in. We re-engaged him each time. We talked about the patient contact list. Make it as easy as it can be to participate. UAB is a big place, or some of the outreaching partner networks. Parking is an issue. So validating parking, meal tickets. I've had patients that have brought their grandchildren and their children in, and we kind of had the research team not provide really child care, but we watched, their babies while we did procedures. So, just thinking of ways to eliminate those barriers and get them engaged and coming in is just really important. So I think that is it. Thank y'all. I know I'm not the best speaker, but I appreciate your time.
So are there any other questions for Meredith? Well, thanks a million. That was great. And so really do realize that CRISP is a terrific resource, and feel free to contact them and explore what they really have to offer, particularly if you need to do a research, you're planning a research study and you're trying to make a decision whether you're going to hire somebody to do your, be the coordinator, or whether you're going to, rent somebody to be the coordinator. It's often really more efficient, and you'll get a higher quality output by using somebody who's really experienced, who's got a lot of experience, who can help you with a lot of the things that you would otherwise have to be learning by yourself. So we'll take a 5-minute break, and then we'll move on to the second half. So, just one other thing, just before you're leaving. So I think if you didn't have a chance to sign up for the IRB scheduling, please do so with Jean. So that IRB meetings will start next week, and then we also talked about an extra session to catch up for what we lost during the weather holiday. And so Dr. Howard is able to give his presentation on Wednesday, the March 14th, and we'll record it. So if there are people who can't come, because that is the UAB spring break, it'll be available to watch online afterwards. So hope that works for most people.
I have a question. Dr. Chapman. Regarding the renting of the coordinator, because there's no questions, they have to have.
Yeah, and that's all you do if you don't have some funding mechanism.
Yeah.
But it's drug studies, that's
Feel the same way.
if you're trying to do your own service or independent. Yeah, I mean, you have to find or something. You know, I think so vouchers to be successful, they want to do something bigger than they are, but I think we
Obviously, too.
prepare for another. So I think what I would be careful about is that voucher reviewers do recognize that you're saying the same question over and over again. So they need to be confident that what you found before yielded something else. So, if you watch that or something, help you send a message and say, thanks for the bunch of things that really helped me. Here's what it did. You managed to turn it into so much more funding. Give them feedback so they know that they gave you paid on.
And then maybe you're.
The program is not to be private funds. It's to allow you to do something that shouldn't be just because the private funds only allows you to get $50,000 and you need $10,000 more to really do the project. But it could be that something came up in the project that could not have been anticipated and you need to do something additional. The budget's on the side, so you couldn't add that on without having some extra money to do it. And it needs to be something that so it's best used for things where the CPS is charge you something separate. Researchers to do something, but I don't think it would apply to the whole project basically. It would have to be to collect something extra that was inside of the scope. Because it's not, so part of the reason there are these rules is because the senator can provide them, NIH senator provides them anything that reviews how it is spent, and it's pretty explicit. It's not meant to be that.
That makes sense. Thank you.
Thanks for doing this. So we have about 16 people on the line, and I don't ask them to mute themselves unless it's noisy. So if I hear a lot of background noise, you don't have to use your computer to make it. But I'm also monitoring chat box on my laptop, so I'll interrupt if they have questions. These are they're from program institutions, and they're all over. So, and then this group is really broad based, right from postdoc to professors in multiple schools. And actually, I have a couple of associate professors. One full professor. So, if I don't turn off the speaker, and then I try to work around with this thing to, so that people are asking questions, so people can hear. If I don't get there in time, if you can repeat the questions, that would be great. And what language you need to use is primarily for them to hear it, so you won't hear that it's working here, but it is giving them. I'm prepared to use any extra time there is at the end, so I think we told you 20 to 30 minutes, but if you need more time, that's fine. Okay. Yeah. Good. Okay. And they're just pretty interesting. I'm just checking for people here. So also, people that have this learning the CCTS are, so if you can help with the Okay, we'll go ahead and get started again. So the next speaker is Dr. Jeff Edberg, who's professor in the Department of Medicine, Division of Rheumatology and Clinical Immunology. His research interests over the years have been in vasculitis kinds of problems, particularly Wegener's granulomatosis and some other vasculitides. He is the person who oversees the one of the sample processing units that the CCTS provides some support for and uses, directs researchers to, and also the biorepository. So he's going to really talk about both what the sample processing core offers, but then particularly about thinking about ways for building your own sample base if you want to use a centralized biorepository for your study development.
Thanks. So, and I'd like to keep this interactive. So I know you guys have projects that you're trying to develop for this course. So there are specimens involved in that. Maybe we can talk about that and how you might think about what you need to collect and how you process them and store them. I should say that the only reason I got into this now more than a few years ago was actually because I had a need. As David mentioned, autoimmunity, vasculitis, and lupus are the two primary diseases I look at. And we were faced with the need to, we were interested in genetics at the time and the role that genetics play in these complex diseases. So we needed to do a lot of specimen collection for a lot of different patients. These are multi-site studies. How do you do that? These were longitudinal studies. We were collecting a lot of different types of biospecimens. We needed plasma, serum, we needed DNA. We wanted cell lines maybe. We really didn't know what we wanted. We didn't know where we were going to put everything, how we were going to track it. So the CCTS, which was the GCRC at the time, was interested in trying to help facilitate that. And so I built, very selfishly, a facility that would help what we needed to do, and it turned out to be quite useful. And now we work with over 200 studies at UAB, not at any one given time, but we have about 200 open studies on the books that we work with around the institution. We do both specimen processing, so you have a patient in the clinic, you need something processed right then and there, we do that. We also do long-term biobanking, we'll go over that in a little bit. We've got an array of freezers over in the Shelby building that we have available to store specimens. But probably the most important thing is we're a resource. I've got a good group. If you have questions about what you should do or how you should do it, just come talk to us. The CCTS has a monthly clinic that's actually here in this building on the first Wednesdays. It's actually from 11:30 to 1. There's usually someone from my group here for that. You can just walk in, get a free lunch, and ask questions, or certainly email or call us at any time. Don't have to use us, that's fine, we don't care. But we just make sure you do the study the right way and answer the questions that you think you want to answer. And so if you have any questions, just stop me and we don't have to follow the script here. So what is a biobank or a biorepository? So these are really specimens you're putting away for future use. Hopefully you don't just put away specimens, just put them away. You hopefully have a need for them. But you should also be thinking about potential future uses that you're really not quite sure of yet, because you never know where the technology is going to go. So when we started our work in autoimmunity and storing specimens more than 10 years ago, a lot of things we're doing now didn't exist then. And so the needs have changed a lot. And so particularly one of the things that has come up more recently in the realm of DNA sequencing is there are some techniques that require really long pieces of genomic DNA, and traditional extraction methods are not adequate for preparing DNA of that quality. So fortunately, we didn't know at the time that we were being smart, but we were smart in hindsight that we stored materials in such a way that we could actually be able to re-extract new DNA now, 10 years later, that is amenable to some of the newer sequencing technologies that take advantage of these really long pieces of DNA to get really long contiguous DNA sequence reads. So you need to give some thought into what it is you want to store, why you want to store it, and where do you think the field's going, so that you can be prepared many years down the road. The hardest part, as Meredith just went over, is recruiting your participants and doing your clinical phenotyping and collecting your data. If specimens are part of your study, you don't want to run out of specimens. And technology is always changing. You want to make sure you have materials backed away in an appropriate way, that you can take advantage of those materials and leverage all the work in recruiting participants into your study. We make a distinction between specimens used for clinical use, so these are ones that typically go right to the outreach lab or to pathology for reporting. Anything that goes into the patient record, we consider clinical use. You don't want to use us for that. That has to go straight to the pathology. They have all their CAP CLIA certifications in place to handle those. We come into play more in the research use, and these where we're storing specimens for research purposes. This can still be done in the context of clinical trial. We work with a lot of clinical trials, phase 1 to phase 4, where specimens are collected, processed, and then sent off to central labs for research-based testing. They can be standard clinical tests, but it's done for the purpose of the research study, not necessarily for driving patient care decisions. So when you collect your specimens, you got to make sure that you know something about them. You just don't put away a blood or a plasma or whatever it is in a freezer and then say, well, now I've got something in there. You need to know something about that sample, and you need to know something about where that sample came from. At a minimum, for your own study, of course, you should have demographic data, clinical phenotypic data about the patient, whatever it is you're interested in collecting, then any research-based results that you think you're going to be collecting that needs to be associated with those specimens. You just got to kind of think about the data structures that you're going to put together and work with your data team and your biostatistician team to make sure that when you're collecting the specimens that you get all the information that you need about those specimens. This can be a big deal. In our studies, we spend a lot of time just trying to catalog the information that we have. We have over 6,000 patients in our study. We've got a couple 100,000 specimens, and then there's all the longitudinal clinical data associated with that. That's an enormous undertaking. And so there are a lot of resources on campus for people to help pull these types of data together for your study. You really need to think these things through. It's always better to do up front than it is after the fact when you've got an avalanche of data or a big need that you need to satisfy quickly. It's just good to always think ahead of time, kind of plan what you're doing. And I should say, when we talk about biobank here at UAB, this is not a situation where you can just come and say, hey, I need 52 plasmas from healthy males or whatever. Everything that we have has actually been collected by an IRB-approved study. These are all investigator-initiated projects of some kind that has collected blood or materials that we put into freezers. I'll talk a little bit at the end about how we can facilitate sharing and forming collaborations, but there isn't a bank in the sense that you can just go and say, hey, I want this. Everything that we do and we have collected has been done in the context of an approved IRB protocol and is under the control of the PI, whoever collected the specimens. So you also need to think about the type of samples that you're biobanking. I'm going to focus most today on liquid biospecimens because that's what we do, but tissues are important too. You need to understand, are you getting materials from healthy individuals? What's healthy mean? So when I think of healthy in the context of autoimmunity, it means they don't have systemic autoimmune disease, but I don't want to think about the heart disease. I don't want to think about hypertension on these patients. But if you're interested in hypertension, that's obviously an issue that you want to know about. So it's just kind of important to think through how you define your patient base and what you need to know about those individuals. You need to decide what kind of materials you need. As I said, I'm going to focus today on liquid specimens, typically blood, urine, CSF, saliva. But obviously tissues are important for many types of research too. I'll give you a link at the end for a very good tissue group here on campus in pathology, Dr. Grizzle's group. He has a large tissue procurement facility that can help investigators collect, process, and section tissue. And then stool is a big issue now too with the interest in the microbiome. And again, I'll show you some links to resources here at UAB. There's a whole core facility that can help you with collection, extraction, sequencing, and analysis of DNA for microbiome. Here are some of the resources, and I think these links are current. I checked them last week, so hopefully they haven't changed too much since then. So at UAB, in the CCTS, we focus primarily on the liquid type specimens. There's a link to the CCTS website where our core is listed. We can handle pretty much any kind of liquid specimen that you have, and again, we can do both same-day processing, kind of in and out. You need to process right away, or we can also do processing and storing long-term for you and help you manage your biobank. For solid tissues, Dr. Grizzle in pathology has an excellent facility for procuring tissue. Kathy Sexton is the manager of that group. They've been doing this for decades. They're very good at it. They're a great resource if you have any questions about handling tissues or how to process or section samples. I'm sure they'd be happy to talk to you. And then for microbiome work, there's a core facility that has support by many different centers that Dr. Casey Morrow runs. And again, he can help with both how to collect specimens, how to process them. They have some capacity for processing, extracting DNA, and then primarily then the next-gen sequencing for getting the microbiome and the analysis. And there's a whole CCTS core that has a pipeline set up for data analysis for those types of studies. So when you think about specimens, you, again, just re-emphasizing it, you think before you start doing your study, what is it you need to collect and why do you need to collect it? So you need to think about what you need to collect, how the samples need to be handled. Some things that you do with specimens may be very time, temperature sensitive or sensitive to how they're handled in the centrifuge, the speed or the time. You need to think about how specimens are stored. Room temperature 4 degrees, are they frozen? Does it make a difference? Is -20, -80, or does it need to go to cryogenic temperatures? You kind of need to think all those things through. And all those questions are really going to be answered by the types of analyses you want to do ultimately. You may not always know what you want to do, but we can provide some guidelines as to best practices on what you probably should be thinking about doing for most situations. And then you need to think about how you're going to use the samples, how and when you're going to retrieve them. So is this something where you're going to collect a specimen and measure something right away? Or are you doing an assay that might have batch effects where you have to batch everything together? Then you need to worry about storage times and temperatures to make sure that the differences in the time that samples are in the freezer won't affect what you're doing. Again, just really thinking through what it is you're doing before you start the study, start using these specimens. Again, you should always think about what you want to do in your current study, but always think about where do you think the field is going? What are you going to need down the road? The most valuable aspects of our lupus study, we've now had probably over 100 papers that we've been participating in that have used some of the DNAs and plasma that we've collected because we planned ahead. We have a lot of different types of specimens available. We've been able to form a lot of collaborations around the world that have facilitated a lot of different studies. So really think about if you get a valuable specimen collection, a valuable patient cohort, how can you leverage that to help get future studies going and inform collaborations with other investigators, not only here, but around the world that might be able to utilize, work with you on your samples. So here's kind of how we view the life cycle of a research specimen. It goes all the way from collection. There's a lot of details you need to think about. Labeling, that's pretty boring, but it's actually really quite important because if it's not labeled right and the label comes off or it doesn't, or it smears or something, then it's not going to be very useful. We've got good solutions for these things, so it's not a problem, but you just need to make sure you understand what you're doing here. You need to think about how specimens are getting from where it's collected to where it needs to go for processing and storage. It's an issue at a big medical center like this. There are patients being seen all over the place, usually the blood is collected at the patient's convenience, not at the study's convenience, which is the way it should be. But then you got to think about how does a specimen get from point A to point B, and that turns out to be remarkably difficult sometimes because you don't know when patients are coming in. There's always variability in clinic schedules. Patients show up when they show up. Things happen. So you need to think about how you're going to get your specimens and where they need to go. I know that sounds kind of silly, but it's actually a big issue that you really need to think through. If somebody's being seen over at WIC, how do you get that sample over to Shelby if it needs to come to Shelby for something? That's not that long of a walk, but it's a big enough hurdle that it just doesn't happen automatically. And then certainly the processing, storage, and use we talked about already. So for specimen collection, I don't know if you probably talked about IRB already. Obviously, before any samples are ever collected, you got to do informed consent, and the patient has to understand what is being taken and what's going to happen to them. We always advocate for writing your consents in such a way that you don't limit yourself. I've seen some consent forms where they say I'm collecting 7.5 mils of serum. Well, that's kind of silly because if it turns out you really need 8, well now you just said 7.5, so now you're stuck. So keep it broad but fair to the participants, saying you're collecting blood for research uses. Typically, IRB allows you to go up to about 100 mils without in patients that aren't critically ill, without a lot of questions. If you do more than that, you need to start justifying. You might have to do a full review, but try to keep it broad. Don't limit yourself to just 1 tube or 2 tubes. Just say up to a certain number. I think that's tablespoons typically, but we tend to typically try to go from 30 to 40 mils, and the consent form doesn't mean you need to collect that much, but that way gives you the flexibility that if something happens after you start the study and turns out, well, we really need to get another serum tube now for something, then you've got the flexibility without having to go back to the IRB and get another amendment done. And also, again, be thinking about those future studies. Don't limit yourself saying, I'm going to do assay X on these samples, and now that's all you can do. So when we're studying autoimmune disease, what we say is, in our lupus consent form example, we're studying the pathogenesis of lupus and immune factors that regulate the disease process in both lupus and other associated autoimmune diseases. That's a pretty broad consent that allows us to look at other things. Maybe if something in diabetes comes up that might be important that we want to look at. We've actually used that in some of our collaborations. We've had broad consent. We've been able to use the samples for a lot of different types of studies. You need to think about incidental findings, and occasionally we get samples, if you get a enormous white cell count, you immediately start thinking that something might be going on. We usually let the study know about that, but you need to have a plan, what you're going to do if something comes up that you're not anticipating, what you tell the patient or your participant. There are a number of changes that have happened recently in the common rule regarding specimens. This is still somewhat in flux. And I'll leave that to the IRB to sort through with you. But again, just need to be aware of what the rules are for storage, consent, and use of samples. Again, keep your consent form fair, but keep it somewhat broad so you don't limit yourself to just 1 or 2 different types of things. Make sure you write it in such a way that you can take full advantage of the specimens that you've collected from the study. So the use of the specimen obviously dictates the types of collection mechanisms you're going to use, but you need to think about the type and the time temperature requirements. Some analytes are very sensitive to time and temperature, and so you just need to be aware of that. Is the blood need to be put on ice or not? Yeah.
I'm sorry, just going back to the information for the consent. What are the details regarding the demographics the minimum amount of details you require for the biorepository? Because probably we need to put that on the consent so the patient is aware that there's some sort of identifiers, age, sex.
Yeah. So there are 2 ways to answer the question. So from a biorepository perspective, we don't really care other than that, making sure that you get what you need for your study. So it's really up to what the study is going to need. I mean, I would have thought at a minimum you'd want age, ethnicity, gender, race. That's pretty standard kind of stuff. If you need anything more than that, more detailed information about where they live or educational backgrounds, socioeconomic status, things like that, that really is very study dependent. The biobank doesn't necessarily keep that kind of information. We'll keep specimen information, but in many studies we don't know anything. All we get is a tube with a ID on it. So our interested in lupus, and we'll get SLA 1, 2, 3. That's all I know about the sample. Now the study knows everything about that patient, hopefully. But in the biobank, we just have the specimen ID, and we don't study its width, so we know who it's associated with. But we don't have a lot of that information. Now, there are some newer tools that I'll go through at the end where clinical trials now at UAB are using a program called Encore to manage the clinical trials. We actually use Encore to manage all the specimens. There's a biospecimen component to Encore. And Encore can collect a lot of demographic data on patients. It can pull information from the EMR, if you know the electronic medical record number. And so that can be kept in Encore if you want to keep it there, but you don't have to. You can have your own study database. So in our lupus work, we have our own study database that's been built for us, and we keep all that information separate from the specimens.
What's the cost for the investigator?
The cost, so that It really depends on the nature of what you're doing. If it's just getting plasma and serum, that's really easy. So it's a couple dollars a tube. But we do a lot of different things. We can do DNA extractions. We can make cell lines. We can isolate peripheral blood mononuclear cells. Those are much more time-consuming, expensive, and it just depends on what it is you want to do. We try to structure our costs so that it's we're pretty low cost relative to most other academic facilities for investigator-initiated studies. We do a lot of industry-sponsored studies too, particularly at the Cancer Center. And with industry studies, they're willing to pay whatever they need to make the study happen, so we can recoup a lot of our costs and kind of overhead in that way and keep the costs low for investigators. We don't currently charge for storing materials. CCTS has provided freezers in the space, and so that we charge kind of the intake, processing it things labeled and getting stored. But once they're in the freezer, there's no ongoing charge at that point. There's no charge to pull things. If you just want to say, just give me my plasma or whatever it is, we'll pull it and give it to you. If you want us to further process it at that point and make some aliquots, whatever, we'll have a minor charge for that. But the charges are pretty reasonable and not that much.
Do you work with the VA, collaborate with the VA in terms of specimen collection and storage?
Can you repeat the question?
Yes, we work with the VA. Not in a direct way. As you know, they have their own IRB. Everything is done with the VA. But there's certainly not a problem. I mean, if a sample is collected in the VA, from our perspective, as long as there's informed consent, that's occurred and it's been approved. It doesn't matter what's from the VA, from TKC, from WIC, or wherever from an outpatient clinic, as long as it gets to us and we know what we're supposed to do with it, then that's fine. So there's no problem there. So you need to know what kind of, if you're collecting blood, you need to know what kind of tube you need to use. There are a lot of different kinds of blood tubes. Do you need plasma or serum? You need to understand the difference. Remember, serum is a liquid part of blood after the clotting process. Plasma is a liquid part when you have an anticoagulant, so it's not clotted. Sometimes those anticoagulants, typically it's heparin or EDTA. Sometimes those can interfere with downstream assays depending on what you're measuring, so you just need to be aware of those issues and kind of think that through. If you're getting nucleic acid, that could be DNA, RNA. That can come from actually a number of different places. Most typically, it comes from white blood cells. We typically don't like to see serum tubes for those. I mean, you can, in theory, get DNA from a clot, but you're much more susceptible to degradation because that blood's been sat around for a while. It's clotted. A lot of things going on. So we like to get DNA from anticoagulated tubes, typically like purple top tubes, EDTAs. One of the little things to be aware of, not so little actually, is if you're getting DNA, you do not use green top tubes. Green top tubes have heparin, and heparin is a very large charge molecule that sticks to DNA and co-purifies and inhibits Taq polymerase, which is used for almost all DNA applications. So that's not a good thing. So we usually recommend purple tops, or sometimes yellow tops will work for DNA too. RNA is very difficult, it's very susceptible to degradation, and you really need to think through what you're going to do with RNA, how you're going to isolate it, where it's going to come from before you start collecting, because RNA has to be dealt with pretty much that same day. There are tubes that allow you to collect blood and stabilize it, and it can be stabilized in a freezer for many, many months. But RNA, you really need to think a little bit about how you're going to use it and where it's going to be coming from. More recently, there's interest in circulating DNA. In fact, there are some companies now that are trying to develop they call liquid biopsies, where you can actually assess tumor DNA in the circulation through next-gen sequencing. But again, that has some special processing needs, right? So DNA in the blood is going to be very susceptible to degradation. So there are very strict time and temperature requirements when you get circulating DNA out of a plasma sample. It's got to be done typically within 1 to 2 hours. You have multiple spins. You got to make sure you get rid of all the cellular material and all the exosomes that might be in that sample, so you have just pure soluble circulating DNA. You got to get that done, as I say, within 1 to 2 hours, depending on the protocol you're following. And it needs cells. Obviously, you don't want it to clot. If you want to get cells out, because the clots, your cells are going to be bound up in that clot, and it's lost. So you want to make sure you use an anticoagulated tube for that. And again, depending on what you're going to do with those cells, will dictate the type of anticoagulant that you use. And again, we can help you make your decisions on that, depending on what it is you think you want to do. BD has, Becton Dickinson has, on their website, it's a nightmare, but if you there's a link there. They have charts and guides about all the different types of vacuum containers. I mean, there are probably 100 different types of vacuum containers, and every possible variation, different sizes, glass and plastic. One other thing, glass, be very careful with glass tubes. Vac containers do not freeze glass vac containers. They do break, and that is a mess when you thaw them. So just, I think these things through. So if you need cells, again, as I said, it depends on what you want to do with them, which anticoagulant would be best. There are some general rule of thumb here. If you're interested in B cells, you're an immunologist like we are. EDTA is not good for B cells. It tends to kill them. And so if you want to do something with B cells, you collect a purple top, you're probably not going to get very far. So you've got to use a different kind of vac container for that. Time and temperature is obviously going to be very important. You don't want to freeze the blood if you're going to try to get cells out, because those cells are going to lyse in the cause. And time is really important. There are Beckman Dickinson cell CPT tubes, which have a built-in separation gradient that allows you to just pump the blood right in the tube and throw it in a centrifuge, and you can get blood mononuclear cells out in about an hour. They're nice. They're a little expensive, but they do help with collection and getting cells quickly. So I've already mentioned serum and plasma. Again, you need to think about time and temperature. Serum requires usually a 30-minute clock at room temperature before it's spun down. That may or may not be a problem with what you want to do. You just need to think through what you're doing and what the time requirements are going to be. These are the typical serum tubes. One of the things with serum tubes, sometimes you see that little gel clot in the bottom. That's really useful for the processing step because it allows you to get a really clean serum separation. And you can get your maximal harvest of serum out of those tubes. When you use a regular red top without a gel barrier in it, the serum tends to kind of mix a little bit with that clot. You can't get all the serum out cleanly. But there are some studies where even though those clots are said to be inert, they can affect serum in a way that invalidates whatever acid they're trying to do. And sometimes there are clot activators in the samples too, so you need to be careful if your tubes has a clot activator. For most things, it's not a problem, but there are occasions where assays will not be compatible with those types of clot activators. Typically, these are red top tubes. Sometimes they're called tiger tops. Those are those red top tubes with black spots on them. Those are the ones that typically have the gel barrier in them. These are the most typical plasma collection tubes you see, green, purple, and yellow. Yellow is ACD that typically we use for PBMC preps, perfect blood mononuclear cell preps. The cells maintain a high degree of viability in those. Again, green tops are not good for DNA and RNA, so remember nucleic acid, not green, purple or yellow. There's also plasma separator tubes which have a gel barrier in them. Those are again nice because it makes it really easy to spin the sample down and get maximal harvest of plasma out of your sample. But again, it may be totally inert, and you may need to think about whether using that is going to be compatible with what you're going to do. They're typically more expensive tubes. Just depends. It's a balance of convenience and time and assay date. So DNA, we talked about a little bit already. I'll reemphasize again, RNA is very susceptible to degradation. You cannot collect the blood tube and let it sit around and expect to get RNA a couple days later. That won't happen. The RNA will be degraded. DNA typically is pretty stable. We get in our multi-site studies, Friday clinics happen, and if that's when your patients come in, you got to draw them on Friday, and we get the blood sent to us. We get it on Monday, assuming FedEx doesn't screw up. And as long as the blood's not been frozen, usually the DNA is going to be fine. Your yield's going to suffer a little bit, but from a tube of blood, you're going to get a lot of DNA, so it's usually not a problem. But RNA, you got to work it up the same day. You got to do something the day that it's drawn, usually within a couple hours. There are patch gene tubes. I'm sure most of you have heard of those. That has a proprietary stabilizer in it that when you draw the blood into the tube, you mix it, and it actually lyses the cells and stabilizes the RNA. You can then freeze it and keep it in the freezer for, I've had one study where we collected RNAs up to a year and then went back and extracted them in batch, did our analyses, and they were fine. The only issue with those tubes is the cells are lyse. So if you need to separate out, if you want RNA from B cells, you can't do that at that point because it's whole blood. It's going to be whole blood RNA. So it really depends on what you're doing. Again, I'll show my immunology bias as an immunologist, but those of us who look at the immune system, B cell is not a T cell. So if you're looking at the transcriptome, they're very different, T cell from B cell. So if you do whole blood, you just got to recognize this is whole blood, so it's a mixture of all the different types of cells that are in the blood. In particularly in disease processes, those cell numbers will vary a lot, right? More patients are treated with different agents. Anti-B cell therapies are very common in autoimmune disease, so maybe they don't have any B cells anymore in the circulation. So you just need to be aware of the composition, too, of what your blood is if you do have whole blood RNA. There are also reagents you can add to blood or cells. One of them is called RNA later. Again, it's something that lyses the cells and stabilizes the RNA and allows you to freeze that RNA, freeze that sample before you extract it later. Those are good, but you have to do it that same day. So you have to get the cells separated or the blood in a state where you can then add the RNA later and get it frozen. You can't let them sit overnight. They have to be done the same day. As I mentioned, where the RNA comes from is important. You just need to think that through. So if it's whole blood, you just got to recognize this is whole blood, and you're going to have all the variability of differences in cell counts and different representation in different types of cells. One of our studies, we're actually separating out B cell subsets and getting transcriptome analysis from RNA on the B cell subsets, but that requires us that as soon as we get the blood, we get it back to the lab, we separate them out, do flow cytometry to sort different cell subsets, and immediately isolate the RNA. That usually makes it very late night, but that's what you have to do for RNA. So I mentioned this issue here, labeling, transport, and processing. So labeling, you need to make sure that if you're processing your samples, the labels won't come off in the freezer. Most standard Avery kind of labels will not last in the freezer. And handwriting is not good. Usually most people's handwriting is not legible on little small tubes. And the ink will run. We've got in the CCTS a whole fleet of label printers where we can print out labels for studies. And they're very stable. They won't come off in the freezer. They won't come off at cryogenic temperatures. Just to make sure you're using labels that are appropriate for how you're going to store your samples. Storage. So you got to remember, if you're freezing samples, if you're using a -20 freezer, most household freezers, they cycle their temperature. That's why they're frost-free. It's really bad for specimens. You got to make sure that your sample is always going to a non-cycling freezer. Most all -80 ultra-low freezers are non-cycling, so those are ones that will just keep your samples at -80. If you use a -20, just make sure that it's the kind that builds up the frost. Also, make sure people understand your people understand the difference between a freezer and refrigerator. You'd be surprised. We have a lot of students that help us out, and I've been surprised sometimes they get confused about refrigeration and freezing, and what's the difference between a refrigerator and freezer. So just make sure it's very clear where it's supposed to go. Be aware of freeze-thaw cycles. Most analytes can go through a couple freeze-thaws. Like DNA, for example, you can do a couple freeze-thaws, but every time you freeze and thaw it, you're going to be changing it and altering it in some ways. Just to be aware of what that's doing to your materials. We usually recommend that you aliquot particular plasma serum out into aliquots that might be single-use size within reason. You don't want to get 52 aliquots for every tube of blood. That would be kind of crazy. But you want to just make sure that you kind of think through how you're going to use the samples and make sure you get a couple that are ready to use, single use, and maybe a couple that are larger that might get 1 or 2 thaws. You need to think about how you're going to track your samples. We have a way in our lab. We have a full computerized database that keeps track of every single aliquot. That may or may not be necessary for your own study. Just depends on what you're doing and how many you're going to have. So there are a number of places where deviations can occur. We've seen pretty much everything. We get tubes that come in that are not even identified. They just have Ken on or something, just like, I don't know what that is. Make sure your samples are labeled so you can identify what they are. Make sure you're using the right vac container. Make sure that the vac containers get full. Sometimes I know the phlebotomists do their best, but you can't get a full tube. But when we get a tube with just a little bit in it, and there's more ETA and then blood in there, there's really not much you can do with it. It's not very helpful. Make sure you understand what the parameters are for your processing, time, temperature, and centrifugation speeds, and that can make a big difference in how the quality of your sample ultimately as you go forward. This is hopefully common sense, but the results aren't as good as the specimen was in terms of both the collection, the handling, the processing, the storage, and thawing and use. All those things have to be thought through. And the most important thing is to know your protocol. One of the points of confusion, particularly in multi-site studies where you get a lab manual if you're involved in a clinical trial, is the centrifugation conditions, and that can be represented 2 different ways. There's RPMs, the revolution per minute of the rotor, or the G-force that the sample is subjected to, which will be a function of the size of the rotor. And people constantly get confused about this. This just makes you understand if you're given conditions on how to centrifuge a sample, is that RPM or is that times G? You need to understand the difference and know how to centrifuge. Temperature is important too. Time. Labeling, we always try to encourage pre-printed labels as much as possible, so you avoid handwriting. Sometimes handwriting's not, you just have to label things, but if you do have to handwrite things, make sure that it's legible. We tend to use labels, we like to use labels that we call wraparounds. It actually has a frosted part that gets printed on, there's a clear part. The clear part actually covers the part printing on it, so the printing can never get smudged or wiped off. In the CCTS, everything we do is barcoded. All our labels have a 1 or 2D barcode on it that allows for unique sample identification. Every single aliquot we make has a unique barcode identifier on it. And again, you got to make sure your labels are appropriate for storage conditions. Some of our particular cells you're going to be storing in cryogenic temperatures, liquid nitrogen, that's about -190 C. You need to make sure your labels will stand those temperatures. And one of the important things, this is, I know, hard in the clinic, because sometimes in large clinical trials, you may not always know all the information about the sample that's being collected and it has to get processed right away. But once the sample's frozen, it's really hard to relabel because it gets wet and the labels are adhesive, so that doesn't do well with wetness. So you really got to know everything you need to know about that sample before you put it in the freezer and get that label on it. It is, in theory, possible to relabel frozen tubes, but that typically doesn't work very well and it's very time consuming. Types of freezer options, as I mentioned, -20 freezers, you got to make sure it's non-cycling, which means that it's not frost-free. That's good for short-term storage of samples, but we recommend that you usually use ultra-low, -70, -80 freezers for long-term storage. And for cells, typically you're going to be storing those cryogenically if you need to get viable cells out. The CCTS has all these types of freezers available to help. We can certainly advise if you need to buy a freezer. There are certain ones that we like. That's just our preference. But some of it's based on facts and preference. Also, need to think about how you're going to organize your samples. I've seen literally freezers where there are styrofoam cups with tubes in them. Some are standing up, some are upside down. They're all over the place. And just kind of throw it in your shelf, and that's how most labs organize their freezers. That's fine, because that's what you want to do, but the way we do it is on the bottom there we have racks and boxes, and every sample we have is in a box, it's labeled, and we track every single aliquot in that freezer. Now, if you just have a small study and you don't have that many samples, you can maybe track at the box level, but you need to have some organization to how you're storing things and keep track of where they are. We can certainly help work through those types of issues if you want to keep your own samples, or use us, we can help you do it. So freezer options, you always need to think, do we need another freezer? Increasingly, there's a lot of interest in green labs. There's a whole initiative here at UAB now for sustainability. And there's actually a sustainability officer. The first thing you should always ask is, do I really need another freezer, or are there other options on campus? CCTS has a large freezer capacity that we invested in 8 new ultra lows last year, which are still available. There's a lot of room. We're not interested in just storing the junk, because there's a lot of junk in freezers. But if you have a collection that's valuable that's being used, we're willing to work with you to try and help you use our storage space, not to buy new freezers. Freezers are expensive to buy, you have to maintain them, they use a lot of electricity, so we can use an existing resource to try to do that. And we're in the Shirley building. The freezers that we bought actually last year, it's a new type of freezer. They actually don't even have compressors in them. It uses something called a Stirling engine. These are the types of refrigeration freezer units that are used in space, in the space station. They're very environmentally conscious. They use a fraction of electricity. They generate very little to no heat. Most freezers generate a lot of heat as those compressors run constantly. So these are really nice freezers. They're a little bit more expensive, but if you're thinking about new freezers, we do need to start thinking about sustainability and some of those issues related to how much energy, because it's not just electricity it uses, but all the heat it puts out, how much cooling do you need in the room to do that heating. You need to maintain your freezers. Again, hopefully this is common sense, but obviously the CCTS, we're very rigorous about this. We have 24/7 monitoring of all our freezers. We have a redundant monitoring system built in too. There are daily logs, everything. Freezers are on backup power. That's something you can do here at UAB pretty easily. Make sure all your freezers are always on backup power. Those are usually the red outlets. If they're not, you can get maintenance over to do that for you. Make sure your staff is trained both to handle these specimens and to know how to use the equipment and maintain the freezers. So specimen management, again, this is just tracking your specimen. What do I have? Where is it? What's happened to it? In the CCTS, we try to manage full life cycle, so we know when it's collected, who did it, where it got stored, and then once it gets pulled, we track when it was pulled and who pulled it, so we know that it's gone. Sometimes we have studies come back and say, hey, give me these plastic, we already took them, and we can show when they took them and who took them. As I mentioned, we use a program called Encore. This is a clinical trials management system that UAB is now implementing on campus. It's web-based. It allows us to barcode everything and track every single sample, every aliquot in the freezer. We can do shipping logs and specimen tracking. We can help you with your studies using this system if you want us to help you. This is just an example of a screenshot. The way we think about specimens at Encore is you have a patient, and then you have an encounter, which is usually the visit, and you have the specimens assistant. As you think of kind of building out a tree, we keep track of all those relationships so we know which visit each specimen comes from and what kind of tube those specimens came from. This is another screenshot showing some of the information we collect. For DNA, for example, we have custom fields that we've made where we keep track of quantity and some of the quality QC measurements we do for DNA. Associated with specimens, but this is all specimen level data. It's not patient level data. This is not a place to be keeping your clinical data for your study. This is really all focused on specimen data. It allows us to manage locations. It's just a list showing every sample, as I mentioned, is tracked down within that box as that grid. We know which position in the grid each sample is in the freezer. There are some software options that are more amenable to individual lab use. Encore is kind of an enterprise solution for a large group like ours, but for an individual lab, it's a little bit of overkill. There are some standalone software packages. I'm an advocate of FreezerWorks. I used to use that. It's a really good freezer management program. It allows you to track all your samples down to the specimen level. Freezer Pro is another one. I know there's one group on campus using that. I don't have any experience with that directly. Open specimen. Some of you remember CA Big, that NCI initiative to try to get web-based data management tools that kind of floundered. But there was a specimen management tool they built called CA Tissue that was taken over by a company in India, and it's called Open Specimen. It's free to use at one level. If you want support, you have to start paying, but at least initial use is free if you want to try that. And then there are laboratory LIMS systems. That's usually overkill, but they can also do specimen management. Options that are not good, obviously, are just paper records or Excel spreadsheets, because they tend to sometimes get sorted funny or get lost or not maintained. Really think about how you want to manage your samples. Again, we're here to help you, so we can help you set up a system where you can just use our group. Now, annotating your samples with data, as I mentioned, you want to make sure that if you collect a sample that's actually got some use, that means it's associated with somebody and there's clinical information available. So one of the things about Encore is, since we can't have the electronic medical record number as part of the system, it can actually link to the EMR. And one of the things we can do now is we can actually link to I2B2, which is a shadow of the electronic medical record system here at UAB. It allows you to, at one level, run queries about how many potential patients are there for a given condition. Has the informatics group talked about I2B2 yet? Or?
Not yet, but we'll...
You'll hear about this more in the future, but what we're doing now with all the BAS specimens that we have in Encore in our freezers, again, they're all collected by studies, so they're all investigator-initiated studies, but if we can get the MRN, they can be discoverable in an anonymous way. So if you were interested, I need plasma on patients with type 2 diabetes or whatever, if we've got the MRNs, that information is discoverable, and you can find out, well, geez, somebody's got a collection of 150 patients with type 2 diabetes where they've got plasma. It won't tell you who the patients are, but it'll tell me then that, hey, there's somebody who wants to potentially find out about the specimens. It's really a way to form collaborations. The specimens belong to whoever collected them, so it's not like I can just give them away. But this is a way of helping facilitate collaboration. And ultimately, that's why we're here, is to both do our research, but also try to build and grow our research programs and form more collaborations. And this is a good way to kind of anonymously advertise what it is you've got collected in the freezer to see them if you have other people with other ideas that might be able to use your samples and work with you on developing the research projects. As I mentioned, the first Wednesday of each month, today, here from 11:30 to 1, there's an open clinic. There's both biostatisticians here. Someone from my group will be here. There's also someone from the clinical research unit, which is available for seeing patients, research patients. You just come in, ask any question you want, and lunch is provided. Here's some more contact information, some web links. Our primary lab is over in the Shelby building, where my office and research lab is. We also have a lab up in Jefferson Tower in the clinical research unit on the 15th floor. That was primarily for clinical trials and clinical studies in patients where samples can be worked up very quickly. That lab also has access to the hospital tubing system, so we can have samples tubed to us oftentimes from the clinic. That can work too. And then we have a freezer room in the basement of Shelby where we've got about 12 different freezers that are available for use. And then there's some links here for information about specimens. If you're shipping specimens, you need to know about IATA. There's a whole online course for getting certified to ship biospecimens. Since those are hazardous materials, you need to have certification order to ship materials. There's a link to do that. UAB has an online course that you can do that. And the last one is a nice link again about the different types of blood tubes and what you can do with them. It's from a company, but it's pretty good. That's it. Any questions? Okay.
Have you ever had a situation where the investigator is no longer at UAB, either they transferred or whatnot, and they still have specimens, and what happens with those specimens in that case?
Right. So that's always complicated. I guess the answer is it depends. Legally, actually, the specimens belong to the institution, not to the investigator, and that's gone to federal court, and there's no question about that. Investigators may want to argue that. They can't. They will lose. So if the institution says they stay here, they stay here. We've not, unfortunately, not had that problem. Usually, UAB is a very collaborative place, and there's somebody who's still here who's using them, and so it's really not an issue. We would just have somebody else become the PI on that study, so to speak. We have had people who have retired or left, and there have been a couple times when they said, "We'll just destroy the specimens." Well, we don't want to do that, but that's what an investigator wants to do. That we always try to make sure that if there's a use for the sample, we can find somebody who can use them. But ultimately, the use of the specimens is dictated by the consent form said. So that goes back to the early comments. Make sure consent forms are written in such a way that you don't preclude potential future use of your samples that might help future studies.
It's a real practical problem that software changes over time too. And so I'm curious, Encore, I don't know exactly when we started, but there must have been some major conversion of your storage information data to bring it up to Encore. And how do you think about well, for investigators who are trying to decide whether they're going to maintain their samples in their own freezer, in their little racks of tubes, or whether they're going to give it into an organized system, I think one of the things people worry about is whether they'll, will the information they need really be available 10 years from now. And my own experience is that it's harder for me as an individual to keep up with that stuff than a centralized facility to do it right. So I advocate using the central facilities. But anything you can say that will give people confidence in the long run availability of their materials, and also just what the process is if somebody wants to use some samples that are being held in your storage, but they are really under the control of some other investigator, how you manage that interaction in a way that supports both investigators in a reasonable way.
So I guess the first question is just the maintenance of software. I mean, software does change, it needs to change, right? And technology changes. So when we first started doing this many years ago now, the software tools were nowhere near as sophisticated as they are now. We've gone through a couple different migrations over the years. Encore is a very dynamic program. It's a very dynamic company that supports it. It's a very large user group. There are almost about 2/3 of the cancer centers in the country use Encore. About half of the CCTSs are now using it. There's a very large user base. We're very active in the biospecimen component of Encore. Our group and a group in Indiana, the two largest users, we've actually helped them rewrite the program to make it better. There are always upgrades. UAB is paying a lot of money to maintain this program. Hopefully that will continue. There are ways to export data out. So if we do need to get data out into some other database or some other form, everything can be exported and made available in delimited files. So you got to make sure that whatever you do, that you can get things back out in an easy way, because things will change and it will at some point probably need to migrate to a new platform. Now, in terms of facilitating use of samples, we have in a lot of cases where we facilitated collaborations, because I don't know every sample and every study that we work with, but I have a pretty good sense of what we're doing and who we're working with. And when people ask me, I'm looking for whatever, I can usually point them in a direction to an investigator. And again, UAB is a pretty open place, and people are usually willing to collaborate. If you're down to your last DNA sample on a really precious patient population, well, that may not get very far. But typically, as long as there's enough material in the freezer, people are pretty open to collaborating here, even collaborating with people outside the institution. And so that our goal, particularly with the I2B2 linkage now, is to have specimens be discoverable in a way that respects the ownership, so to speak, of those samples, so that it really still was under the control of the study and the investigating team that collected the study. It's not under my control. And ultimately, it's under the IRB control, right? Because you really can't use specimens for things that haven't been consented for. But it really is a discussion between somebody who wants samples and whoever collected the samples to understand whether the collaboration can be formed or not. Yeah.
So for how long do the samples remain viable, like serum, plasma, we collect them and store them?
Yeah, so the long-term viability, so that really depends on the sample and how it was handled. DNA, we've had DNA in the freezer for 15 years, but it depends how it's extracted, right? So there are a lot of different ways of extracting DNA. There's some really quick, easy ways to do it. Those are great because they're fast and cheap, but DNA doesn't last long term. They're not as clean. We do charge a bit for our DNA extraction, but it's really archival quality DNA. Like I said, we've had DNA in the freezer for 15 years, and they've been fine for next-gen sequencing. Typically, we store blood or buffy coats for future DNA extraction. I'm not sure there's a strict time limit on that. I would say probably 10 years is you're going to start getting some degradation. As long as they're at -80, you're probably fine. Plasma serum at -80 is usually pretty stable. But you just need to be careful if you're doing a study and I collected, 15 years ago some plasma and I've got some fresh ones, you probably need to do some control experiments to make sure that what you're measuring is comparable given that time frame. But certainly, a couple of years for plasma serum is usually fine.
And so for as long as you're keeping those samples, you have to maintain your IRB?
Yeah, so that's actually an important point. So and it's something that we're always active in trying to maintain. In order to use those samples, you've got to have an IRB. I mean, it can be long-term follow-up. It doesn't have to have new recruitment, but you still need to have your IRB open for use of the specimens.
And the long-term follow-up does require submitting an annual report to the IRB still, so you can't build in long-term follow-up with no paperwork.
Right. But it's pretty easy form. It's just box long-term follow-up and no changes and a signature, so it's not that onerous. Okay.
Okay, thanks very much. So I'm a little frustrated because we have originally, we encouraged you to engage in an interactive activity with the program at the beginning of the session, and we had built in a substantial amount of time to talk about it, but it got canceled by the snow holiday, so the non-snow holiday. So anyway, we're struggling a little bit to find ways to put this back into the program. And what I want to do, we only have 7 minutes today. I'm going to take 10 if you don't mind, but then we'll try to build in a little more time down the road. But really, the exercise was intended as a mechanism to get you thinking about how to conceptualize a research project and how to communicate it in a way that would engage others in understanding your excitement for the project. So we didn't give you very robust directions, and if we were doing this again, we would give you more feedback at the beginning. But anyway, so I want to talk through both. We asked you to do two things at the beginning. We asked you to provide two research questions that you were really excited about. So I'm not going to address that very explicitly today, except actually I wasn't intending to, but that's what I really want to talk about. The second thing was to get a sense of what it's like to try to work in a group. Now, for us, the groups were very artificial. We just took names out of the registrants of this program and tried to make it as diverse a group as we could, both in terms of the kinds of academic units you come from, where you come from, whether you're male or female, and what your research interests are, and to ask you as a group to communicate, identify some challenging biomedical issue that you found really important, and stipulate what that challenge was, and then give a brief explanation for why it was important to address the challenge. So what I'm going to do, I had originally planned that we would have half an hour here and we would go through each of your reports for those to give you a sense of what your different 6 groups thought about. And I'm going to send those around because I think at this point, I just would like you to see what kind of issues you felt were interesting. But I want to preface that the instructions were pretty explicit for this. They were vague but explicit. So choose your group's translational challenge and develop within your group a consensus statement about why the challenge was important to address. So what happened, and I'll send the document around so you can see it, was that the challenges were articulated so broadly that it wasn't easy to tell what the challenge was. So in some, it wasn't explicit. So, I'm not trying to pick on anybody because this was a general issue, although there was some variability of how, by my criteria of what success was, how close you came to something that would be viewed as pretty successful. But some, it was, the challenge is diabetes. So that is a huge challenge, but I think to be a useful challenge, it would need to be articulated a little more clearly what within the broad field of diabetes the challenge was. And maybe the challenge is to reduce the incidence of diabetes in our population of a specific population. And then what you were very successful doing, or